In this series of 4 videos, we're going to go over how to calculate allele frequencies and expected genotypic frequencies. By expected, we mean the frequency of each genotype if the population in question is in Hardy-Weinberg equilibrium. In this first video, we go over some background to orient you to our practice problem. You do not need to remember any of these details. Again, they are just to orient you to this example. For this example, I'm going to use my dog, Indy. She's usually fun, occasionally kind of freaky, but usually just fun. One fun thing about her is that she has some bonus toes on her back feet, and these are called hind dewclaws. Most wild canids have 5 toes on each of their front feet and 4 toes on each of their hind feet. Since domestic dogs are descended from wolves, they also have 5 toes on each of their front feet and 4 on each of their hind feet. However, in some large breeds of dogs, such as Great Pyrenees, there is an extra toe on the back, or a hind dewclaw. In Great Pyrenees, there's an autosomal dominant mode of inheritance for this trait, but it varies by breed. And one of the genes found to be responsible for the presence or absence of hind dewclaws is the LMBR1 gene, although there are also other genes that have roles to play. Remember, you don't need to worry about memorizing any of these details. We swabbed Indy's cheek and sent in her DNA to Embark to have it tested. We found out what her breed mix was, we found out about her health and some traits, including the LMBR1 gene, which has a role in whether or not your dog has hind dewclaws. You can see here that her genetic result is CT, likely to have hind dewclaws, which makes sense because she does. In this case, the T allele is dominant to the C allele, and dogs with at least one T allele are more likely to have hind dewclaws than dogs without it. That is, dogs with a CT or TT genotype are about 50% likely to have hind dewclaws, whereas dogs with a CC genotype are less likely. In the next video,